Uh, Hi everybody. Uh, we are going to have the second session on translation. We are going to examine today how the process of translation completes by three important steps like initiation, elongation, and termination. Just to recap, we said translation is a process where protein is synthesized. Protein is made up of polypeptide chain. Polypeptide chain means a large number of amino acids joined together by means of peptide linkage, and that is called polypeptide chain. From polypeptide chain, we get amino acids. This happens in the cytosol of a cell. In the cytosol, there will be a large pool of amino acids, and that will be 20 amino acids are there, and these amino acids are there. And for this purpose, we know already that there is already an mRNA is here. mRNA with its codons has been written. Codon is a three-letter abbreviation to read a particular type of messages. That is the codon. It has come from the DNA on the DNA, and then there will be ribosomes on this one, and then a number of enzymes will be there, proteins will be there with tRNA molecule. All this mechanism, these macromolecules are required. They will have a highly coordinated way of working, and then they will help in producing the polypeptide chain. We shall try to know the fundamentals of these things immediately. So what happens, we discussed yesterday, initiation is a process where the smaller unit of the RNA molecule first binds itself into the mRNA molecule, and then the larger unit also joins with that. Larger unit will have with it the energized molecule of the tRNA, which will contain a particular anticodon. So we know that three triplet letters on the mRNA is called the codon. Triplet letters on the tRNA molecule is called the anticodon. These things we already learned. Now, when this, that is when the larger unit, smaller unit, and the first site called AUG or the initiator site is filled up by the anticodon carrying the methionine amino acid, and that is the process telling that the initiation process is over. When that is done, all the enzymes that is required for this one plus proteins, they all go away and only left with these things. Now this particular type of things we will have in this larger space of the tRNA molecule. We'll have what's called A site. That is the site where the new tRNA molecule, which is called tRNA, containing an amino acid residue with energy in that, is called a charged tRNA or containing energy. Like that, 20 different amino acids may be picked up by the tRNA molecule. To that, ATP adds energy, and then that becomes a charged tRNA molecule. Like that, this is the charged methionine tRNA carrying molecule. It has come there. If you ask me how will it come, which area it comes, how it comes, are all wonderful thing that happens in the cell. We will not be able to know much of it. All the molecular drama, interplay that happens inside a cell is wonderful. We will not be able to discuss that here. Then there will be second site, and this site is called the A site. At the moment, the A site will be vacant in the initiation complex, and this is called the P site. P is the short form for peptidyl. That is called the peptidyl site. And there will be one more called the E site, and this is the site when the last part, that is the tRNA molecule, goes away. So 3 sites will be there. Now already the process of elongation has started, because one of the charged tRNA molecule containing the first one called the methionine has entered into the complex of the RNA where the polypeptide chain is made. That's why it's called the peptide site. It has come and sat here. That is the first thing. When that happens, next will be the entire thing will have to wait to see the second place is filled up, and that is the arrival or amino acyl space. That has to be filled up. Now how it is filled up? Now we got the codon AGA. To match that, we must have something. Can we put A here? A? It won't accept. So when it reads, it rejects it. How sensitive it is, who tells them right or wrong, how the signals are there to say it is wrong and reject it, are all the secrets of the biochemical materials that happens inside. However, for our purpose, I have taken here 1, 2, 3, 4, 5, 6 different kinds of amino acid plus the initiator amino acid, the methionine. So we will be getting a chain of 7 amino acid sequences in the polypeptide way. Like that, it may be 100 amino acids may join. That means amino acid phenylalanine may form 1, 2, 3, 4, 5 times. GGA form 10 times. Like that, maybe 200 molecules, 500 molecules, 2000 molecules, 4000 molecules. Any number of amino acid molecules may join depending upon the instruction on the mRNA. Now that happens. Now once this comes here, we have to get a proper one. Now the proper one for this AGA sequence will be, let's see, the first one should be U, and second should be C, third should be again U. So we get here a molecule which is having the UCU. This molecule comes to the A site. What is that one? That will contain a U to match the adenine, and for guanine you will be having cytosine. For adenine again you will be having U. This type of a tRNA molecule comes here and then sits down on that. On the top of it there will be one amino acid molecule. Of course that we have named as arginine. Let me call it number 2. So this is number 2. It has come and sat here. So once this happens, somehow signals will change, molecular attraction will be there, and this molecule, that is the number 2, which has arrived on the A side, will now get into this one. They get a connection. They will be connected by means of the peptide linkage. Peptide linkage means the amino group of one and the carboxyl group of the another one will join together to form the peptide link. Now what happens is, when the peptide link is formed, this tRNA molecule dissociates, and that will now come. This UAG has gone, this has gone, this has gone. So all this will now, they move to this site. That is the one without any residue of the amino acid. Now this immediately what happens is, due to some kind of an attraction, this moves away into this, moves away here, and then it comes here. This has gone. So now on the A side, we have, this is number one, this is number one, number two. Two amino acid molecules are added, that is one peptide link has taken place. Now this site, P site is vacant and A site is full. At this point of the time, the entire ribosomal sequence moves one codon away towards 3' end. When once that moves to this, that means this particular complex that is there, that now all this will be translocated to this position. That means that is A, G, A, U, C, U containing a tRNA molecule with one of the amino acid residue, number 2, and then the other one, the first one that is earlier it was there, that will be number 1 will be there, and this kind of movement goes on, and that kind of movement from one place to other place is known as translocation. So translocation moves the amino acid chain, that is 2 amino acid chains are here, moves into peptidyl site again. This is ready to do one more peptide link, and to this site, now this particular combination comes, and that will be, what I do is, I will just move this, the entire unit of the mRNA, the ribosome, to cover this place, and this entire thing will be now, the entire thing will be A site. So one step it has moved, A site, P site and E site. In the E site, what happens is, the moment it comes, that is, this is translocated from here, this will now disintegrate and it goes away. So this type of rotation and the activity will be going on here. Now let's take one more example and study. Now CCU is there. For CCU, what is that we have to get it? For C, we must get a G. And again, we must get a G. 2 G's are here and A. So we get G, G, A anticodon. And this is the one that will now move into this place, into this A site. When it comes to A site, that is the GGA, and that will contain what that we have moved now, that is proline. That is number 3. We shall call it number 3. So 3 has moved in, and that is the proline. So that means the molecule, the tRNA molecules which are charged will move one after another as the mRNA keeps moving one above the other. So as I said, we have taken 7. We imagine that now this again, what happens here, what happens is, this particular molecule again they get the molecular attraction, and then this will link with this one. And that will form a peptide chain. When the peptide chain is formed, this goes back on the A site. So on the A site, we will have now this, the entire thing bounces back there. That means you will have here 1, number 2, and then you will have number 1, like that. So this has gone, and this particular thing now moves into the E site. Now P site is vacant. So now 1, 2, 3 amino acids are added, and this particular thing now moves towards, or the entire thing moves to the next level. As it moves, this particular site also moves now. What happens now here is that now this is there. Now the mRNA molecule, the RNA molecule is now from here. It has moved to this place, and this also has gone, and this P site and other things have gone here, and this place has gone, and now this is now here, and now the ribosome is like that. That's the ribosome. Now in this ribosome we have one A site. Now we must get the P site and we must get the E site. So this is the P site. In the P site now this moves to P site. See, that's the beauty of the translocation. When this happens, this moves to P site. Now here you have C, C, U, G, G, A, that is the anticodon, and this is the tRNA molecule, and that will now contain how many? 1, 2, 3 amino acid linked by means of polypeptide bonds. Now this is gone because this has moved to this place now, taken, and this is gone. This chain is also moved to this place. Now A site is free. Now again the movement goes on. As this moves, this becomes the place for this U, you get A, and then like that. It keeps on going. As it keeps on going, I only mark the first one here, like that. As it goes on and on and on, now we ultimately get a long chain. I will remove this, and then we will write here the first one and the subsequent ones, second one, third one, fourth amino acid, fifth amino acid, sixth amino acid, seventh amino acid. They are added here, and this particular thing will now move away to E site. From the E site it goes away. Now all these things have gone. Now this is moving, and the mRNA, that is the whole complex, moves to the last site, and the last site contains, now the last site comes to A. So what we do, we move the entire thing to this site here. That is the mRNA has come here, and this is the A site, and in this particular site we have already done. It has gone. All these things have gone because we already have a chain of 7 amino acids here. So now here in this particular thing we have this will remove one, first one, second, third, fourth, fifth, sixth, and seventh one was there, and seventh one was maybe UUA, that is the transfer thing. Now this is there. So this has come now. When this comes to UGA, there is no matching tRNA for this, and that's called the stop codon. There are 3 stop codons. One is UGA, UAA, UG. When it comes here, there is no matching anticodon from the tRNA molecule. Therefore, what this cell has done, it has produced a protein, and that protein comes and binds onto this end, termination. Now that marks the termination. That means there is no more tRNA with a definite type of the amino acid. It closes. As it happens, now this particular chain that we have already taken, we have taken now, containing 1, 2, 3, 4, 5, 6, and the first one, as I told you earlier, the initiator one will be there, and that is the methionine. That is a long chain of 7 amino acid polypeptide chain has formed. Like this, you may add at least 100, 200, 300, 600 or 2000 or 8000 or 10,000 amino acid molecules, and then that will be called a polypeptide chain. So now with this, the elongation requires energy, and that energy is supplied by means of ATP and also by GTP, ATP and GTP. When the elongation is going on, a large number of enzymes will be there to assist the translocation and the elongation enzymes, and then will be what is called elongation factors, 2, 3, like that. So all this together will form the complex mechanism called the elongation and termination. Now we come to the last part, and the last part of that is called the modification part. Well, I am not a biochemist, nor I am a cytogenetist or anything of that sort, so I will not be able to tell you much on the modification part, except to tell you only one information, that is, the scientists have estimated that the human body cell may have about more than 80,000 types of proteins. 80,000 to maybe 4 million types of proteins are possible in the human body. That means the first one, what we get is called the primary one. It will have small fold. Then this will have a number of folds inside, forming a complex molecular structure. Like that will be difficult, and that's called the tertiary, primary, secondary, tertiary, and maybe quaternary. Bending takes place, and that becomes a complex polypeptide chain, and that is often called the protein. And this protein, now simple protein, to that modification may take place. That means different biomolecules may be added, part may be removed, and so many, or it may be inorganic molecules may be added, vitamins may be added. All this will happen in the modification. So in our body, every time, in every minute, large number of chemical changes will be happening, which is very difficult to say how many will go on. And such changes will keep on going, and the molecular drama is completely coordinated by means of DNA. Without DNA instruction, nothing can happen, because DNA sends the instruction to mRNA. mRNA will help the construction of the proteins. You know that every body in every part of the body contains the proteins. Even the cells, what we have, will have the proteins. So protein synthesis is a very important and very, very complicated biomolecular process that happens in the cells. Thank you.